camera.
All right, let's get started. Thanks so much, Jamie, for joining. It's really great to have you on our first guest on the podcast. So
excited to be here, Finith.
Yeah, absolutely. Yeah, we've been doing each other for quite a while, right? Like 10 years, something like that.
Yeah, I would say, I think beginning of 2015. So we're a little more than 10 years on.
Absolutely. Yeah, it's been a great 10 years.
It's been quite a ride. 10 years ago seems like a lifetime ago. So absolutely.
Yeah, absolutely. Yeah, we're still getting out of our postdoc, but yeah, no, I wanted to have you on. I think you have a lot of great perspectives, and I really like to hear, from my perspective. I have an immunology question, I go to Jamie. So if I have a question about immunology as a chemist and a background in chemistry, that's my, I really appreciate your taking immunology and boiling it down to where a chemist could understand. So
I feel the same way about you in computational chemistry. So right back at you.
Oh, thanks, Jamie. Yeah, no, you want to start? Maybe we start with the introduction and then go on your background and how you got to where you are, kind of thing.
Sure. Yeah, happy to introduce myself. So Jamie Rice, my background is, I've always really been interested in biology and systems biology, I think just by nature. And, that led me to really immunology is the discipline that I felt most strongly aligned with and really enjoyed, researching. So that started in earnest for my master's degree and then really for my PhD. So I did a lot of the adaptive side of immunology, which is effector T cell development and how your body essentially just adapts and learns, to develop its own unique immune system based on the environmental stimuli and then the genetics that you have. And basically what are the kind of the environmental cues or physiological cues that guide that process. And I really studied T cells as an independent kind of microcosm of that larger adaptive immune response. Did that, studied T cells, and then got my PhD in 2014. But that was about the time that people might remember that oncology and immunology were really starting to come together. And this was the advent that we were seeing anti-PD-1s and checkpoint inhibitors really showing incredible promise in the clinic, really changed the way I think that we treated cancers for the first time in more than a generation, really. We had just been really thinking about first kind of broad spectrum chemotherapy. Then we went to precision oncology medications, for specific oncology targets. But 2015 is where you really saw immuno-oncology start to take off. So I did some of that. I did some vaccine research, some immuno-oncology research. And that really is what led me into my first position where we met each other. Finith, we were working together as postdocs, obviously, at Harvard Med School. That was a time where although everyone was really interested in checkpoint inhibitors, we also thought that maybe agonizing a target and really activating the immune system rather than just taking the brakes off might be an alternative approach to really speed up and help certain people that had refractory cancers. So there was a really, really cool target called STING that I think had the therapeutic promise. Had shown some really strong preclinical data on that, but there were no good small molecule agonists. And so we had right there a great challenge in front of us, right? Discover and develop a novel STING agonist that could activate the immune system for immune oncology, for broad spectrum like solid tumors. And I think that led us to what became a clinical asset, right? SNX-281.
Absolutely.
Yeah.
One thing I've always appreciated about you and your in immunology is it's like for every target, it's just the way that you modulate it. So if you agonize or antagonize something, it can be applicable to some type of autoimmune disease, or if you say to do the opposite, it's it could be a cancer immunotherapy type of treatment. Do you think that's generally true, or is that that's just an impression I have in my head of chatting with people? But I know immunology is so complicated that goes off in many different directions.
I think it speaks to the connectedness and the systems aspect of immunology, right? And there's no part of immunology that exists in a vacuum. It's all it's all interconnected. And I think that what you're saying right there absolutely speaks to it. So modulating one aspect of the immune system will have broader implications on the whole immune system. And so if you have hyperactive immunity and autoimmune disease, then that's the same type of thing that you might get some therapeutic benefit from antagonizing it. So actually, I would say right now the field is much more interested in antagonizing STING for autoimmune diseases, even than I'm seeing research, although there is still ongoing research for agonizing it for immune oncology. But absolutely right. The same target can be modulated in different ways to help with different diseases and different immune systems that are out of balance.
Sure. Yeah, absolutely. Yeah, it's always been another thing with immunology for me that you really taught me was like it's like the exception can be the rule kind of thing. It's like it's always the exception is the most important important part of it. It's like I remember coming to you, you're like, I'm trying to understand whatever is in immunology here, and it's like oh no, that's actually the exception, but it's the most important part. And I was like, oh man, yeah, it's really helped me. I really appreciate your help over the years and really kind of boiling it down. Like I mentioned. So
Yeah, I think that's true for all science, right? Like it's kind of sometimes finding those exceptions to the rule are most exciting, and like that's where the biggest breakthroughs come from because you're stuck in a dogma that limits you, and then by understanding it in a different context, finding that exception is where you make a breakthrough.
absolutely. Have you found that in your day-to-day job today, or is it kind of I know you don't want to go into the specifics or anything, but like as a practicing immunologist, I'm more that kind of thing.
No, absolutely. Because I think why it's so important to me, right, is you're going to understand a target and you just have to read what other people have written, right? And many times you're forced to organize information into both, verbal, but also then these pictures. So you see these pictures of systems, this affects that, therefore changes this. And they're always with arrows and they're always one direction or two directions. And you think of it as this very simple, like, dogma of like, oh, this goes up, this goes down, this gets up, and then you're good. And obviously, it's always more complicated than that. So I think keeping your mind open that is, it's a good place to start. But every model is wrong, although some models are useful. I think is the famous quote.
Yeah, that's a great quote. Yeah, I really like that too.
That applies to immunology as to anything else. I think, and I think about that. So where's this model is wrong? Probably by necessity. But and then how do I make sure that I'm not missing something? Because I'm stuck in some narrow view or narrow perspective of the system.
Sure. Yeah, absolutely. Yeah, I think it's super easy to get into that type of perspective. Oh, one thing before we get into the detail, I was wondering what you thought about the Nobel Prize and that you were like, what do you think about this?
Yeah, so I was pretty excited. I'd read a lot of the paper from all three of the people that co-received that prize. And certainly my PhD was like a subset of Tregs called TR1s. It's a way we just induce them and so we can study them in labs. Was super excited. I mean, there's so much more to do in that field. And actually cellular immunology, I'm thinking like adaptive essentially like grafting T cells or actually using cells themselves as a therapeutic, as in is something that's in its infancy. We're starting to do it with CAR T cells already, but I think we're going to see more of that in immunology too.
Oh, you think so?
Yeah.
Actually engineering and then taking patients. Dude, would it be like like with CAR, you take patient cells, immune cells out? Could you have like a more general therapy, you think, or would it always be you take a patient cells, that are like you do with CAR, you take T cells out, you reprogram or essentially target them towards a specific antigen and then reintroduce, and then you see the expansion and then they attack whatever is expressing that antigen. But do you think it would be the same type of thing, or would it be like hey, you could make a generalizable cell? I guess that would be hard because you probably get your body would attack somebody else's cells pretty easily, I would think.
You just highlighted, I think, all the major challenges in the field. So autologous adaptive T cell transfer, taking your own cells out, reprogramming them and putting them back in you, is what we've done. That's what CAR T cell therapies are right now. So you avoid all those like graft versus host or like cross-reactive responses. The problem is you can't do it very fast, right? It's very expensive. It takes a long time, and it's just not scalable. So people are obviously are very familiar with this problem, and so they're taking new approaches to really address the scalability aspect of that. The proof of concept is there. I think there's three or four now. I know there's at least three marketed cellular drugs right now, right? So it's almost a process rather than an individual drug. But CAR T cells, what people are doing now, and actually some really good breakthroughs coming in, and essentially using CRISPR and adenoviral vectors essentially so that you can reprogram your cells in vivo, never having to take the cells out to achieve that.
Interesting. That's so you could just reprogram it without even having to remove, and then you have to culture and then you have to put it back in. I remember reading the first CAR, one of the patients, I think it was in a Carl Jung paper or something, that they died from the cytokine storm that came, but they were able to
therapeutic stuff. There's like for sure that's in oncology. I think sometimes, I don't want to say that happens, it sounds kind of crass, but Yeah, you're dealing with kind of like some unpredictable.
Yeah, no, it was it was an end of life. A lot of
Yeah.
very, very late stage patients that had really no hope otherwise. And they saw, I think it was like 4 out of 5 patients or something completely recovered.
Exactly.
I remember I read that paper. I was shocked. I was like, wow, this is seems to be a magic bullet, in a lot of ways that, you think, I don't know, from a chemistry background, I think like every drug has to be a small molecule. Right? That's what I thought when I was in grad school and stuff. And then you think like, wow, it's not even big. It's even bigger than proteins. It's even bigger than the antibodies. You could put a cell in there that's engineered, or you can give some other way to, so I think a lot of these alternative therapies are very, very interesting. And, like it's to where you have to kind of expand your mind a little bit on what is a drug and what's not a drug.
Couldn't have said it better myself. Yeah. And I think that's really where you're seeing more and more, like, is that although small molecules will always be an important part of therapeutics, right? For all the reasons. And they have a foundation, they're great, but peptides, antibodies, engineered proteins, adenoviral vectors, antisense oligos, right? mRNA nanoparticles, cells, adoptive seeds, CRISPR, like, it's just expanding and expanding. I think of the FDA, it's funny. It used to be, right, like small molecules, you would have to get a new drug application license when you wanted to market your drug. And if you wanted, other than a small molecule, you had to get a biologics license application. So BLAs were like the wild west of drugs, right? Because different than small molecules, it required a totally different set of skills to evaluate their safety and efficacy and make sure they were like approved right. But we've gotten so, like, now antibodies and engineered proteins are actually go under NDAs because the whole FDA is like, we know how to do this. It's so like straightforward at this point. We actually run those through the small molecule regulatory path, and then the BLA licenses are almost now more like for like these crazy, AAVs and just things that are a little outside the kind of normal scope of think about therapeutics.
Sure. Yeah. You mentioned peptides because I remember, thinking like, oh no, peptide can't get in a cell. Then you see these great engineered peptides that are, extremely interesting to me because, you could run a quick phase display, see some type of efficacy, and then, know you have a good target.
Yeah.
It really kind of, as a small molecule or somebody with chemistry background in small molecule drug development, it's like, you did all this work. You get to the point of, your compound hits the target, it's pretty safe, and then you run through clinical trials, you get to the end and you find out this is actually the wrong target. So it's like it's a real that was really a warning when I first learned that was a major thing. And it's like, well, how do you get around that? Well, it's got to be easier to hit a target. So that's where I think you get these antibodies, you get the peptides, and I certainly have a lot more interest than I did just coming out of a chemistry background. And when I was training in peptide chemistry, it's like, well, drugs are pretty difficult to make, peptide drugs, and now that's seeming to no longer be the case, which is people have been saying that for a long time, but it seems like just recently that you see many more type of peptide drugs that have come out there. So
yeah,
I mean, at least that's my perspective. I'm sticking to it. Yeah.
No, I totally think that's right, man. There's a central thought process here, right? Like where modularity becomes scalable, and amino acids, proteins are the very first like modular design, right? Putting together pre-established building blocks quickly to make something new as just a general concept, right? This is how like Henry Ford made the automobile available to the everyday man, and it's why protein synthesis is so efficient. But for drug discovery, there's a lot of, you can actually take that modularity and really leverage it. So, and I'm thinking about in the discovery process, it's phage display or ribosomal display libraries. And that now, like, I know this is something you know a lot about and more than me, which is non-natural amino acids. And how do we incorporate those early rather than just in the optimization stage, but even in the discovery stage? So we can explore different
more diversity there, right? With non-natural amino acids that hopefully you could, hopefully get better properties or something. Hopefully you could,
absolutely right.
Model it in some way. I know there's a lot of companies out there doing that, trying to model, can we get the sequence? Can we make it into a drug? Can we get it in the cell? All those types of questions that, I remember grad school is like peptides. No, they're great proof of concept, but it's actual drugs, kind of was, there was some pushback, at least in academia. So, I think it's really awesome because I feel like I might have gone a little bit of a different direction if there wasn't this kind of, hesitation, from a lot of very more senior senior scientists, that it seems to be playing out very differently now, which is, I think a good lesson.
This is deja vu for pharma, for big pharma and for biotech, because I just remember when I was a grad student and or undergrad in 2000, and my boss was like, antibodies will never be drugs.
Yeah. Oh, really? So
that's not that long ago. This is 25 years ago, right? That it seemed to it was a great proof of concept. You can do proof of concept, but it'll never actually be a marketed, scalable drug. And here we are today, and 8 out of the top 10 best-selling drugs are monoclonal antibodies. I think peptides are in the same type of thing. People just thought, they won't make, they're just not, the bioavailability is not good enough like a small molecule. But we've overcome that now orally with permeation enhancers. We have long-lasting peptides. So now, the best-selling drug in the world is semaglutide, right? Which of course is a macrocyclic peptide that only has to go in once a week. So I mean, things change really quickly. So you got to be careful not getting stuck in those dogmas like peptides will never be drugs.
Yeah, absolutely. I think it's it's a very interesting thing to learn. I think as a early stage scientist, as a early career, I would say scientist, is that there are these assumptions out there that just kind of born to be challenged. And I think if I went back and did it again, I would actively seek that out and then write a grant proposal or something against hey, I'm going to change this, or
people who do that are the ones that tend to be very financially successful. So I think Eli Lilly, the most valuable pharma company in the world now, because they challenged exactly that and said, I think peptides could be a good drug.
Absolutely. Yeah, definitely. Yeah. So one of the other things too, I was interested in your perspective, since we've been working together, and there's a lot of for a long time, and there's a lot of tech in pharma that seems to be kind of permeating. I think you have from a biology perspective, a lot of unique insights there that you've seen. Part of silicon. Certainly the thing there was to infuse technology in the pharmaceutical industry, and then do that. Have you seen any what was that like, rather than with that type of those initiatives? Was that more helpful or hurtful, or what were the good things and the bad kind of that I was interested in?
Yeah, I think computational drug discovery, which can mean a lot of things, but broadly, I would say, looking back over the last 10 years that I've really been intimately involved with it, with you, obviously, is, I think that you've seen real contributions in what I would call the optimization space. And I think we're still learning how to leverage virtual screening to really start, right? I've seen a lot more traction with things that were discovered through more traditional empirical means, starting points, but then being able to rapidly optimize them, because structure-based drug design is a real thing. And it used to be a lot of guess, and we get co-crystal structures and you could kind of see it. But now this is where AlphaFold and AI and all those kind of things can just take in a lot more data than a human can possibly process, and I think really aid rapid optimization of molecules much faster than a human, just a medicinal chemist, would be able to. And we've already seen this, right? I mean, this is actually where I think the field is right now, and everyone knows this and is using it, which is great. I mean, and AI plays a huge role in that. Again, it's integrating things like AlphaFold together with generative AI and generative chemistry, right? And those things, and then being able to just rapidly reduce your cycle times, get through more iterations, and I just think optimization becomes much faster. So that's the one place I've seen it already take hold and add value. But the problem of screening is much bigger. It's much more complex in a way. You're starting with a lot less information and a lot more uncertainty. So I don't know. I think eventually we'll get there a little bit, but virtual screening maybe is the next frontier for kind of the silicon-based approach to wet lab science. At least that's my opinion, or what I've seen.
Sure. No, absolutely. Yeah. One thing you mentioned that was interesting is the how do you decrease cycle times? That's been something I feel like progress can't be made without another cycle. They're going to get turned.
Yeah.
Well, how do you increase that speed? Because it's not only, to me, it's not just hey, you can get more accurate. So you can say, hey, these molecules that I made, or these molecules I designed, or whatever, are going to be improved or more potent, or more have better properties, or better permeability, or you name it. But you could also say, I want to decrease the cycle time, so I get more cycles in the same amount of time with, say fewer errors, or fewer kind of packages getting lost, or so one of the things I've been thinking about recently is how do you infuse tech into doing that and it's more like a logistics.
Yeah, absolutely.
Logistics and like tracking and ensuring, like, if you have 3 or 4 reagents that are coming from different places in order to run an experiment, if all of them are not in one spot, then you're gonna have a tough time running the experiment. So that's kind of one thing. Have you seen anything with that, or do you think tech could play a role there, or is it, I see a lot of people that are kind of like not really thinking, not that they're not thinking about that, but it's kind of like there's less acknowledgement. Everybody's pushing towards like, hey, if we make these models better, it'll make it go faster. When, hey, lost 3 or 4 days for lost 3 or 4 days in shipping or something that could also be kind of cleaned up, kind of thing. At least that was what I've seen and I've had to deal with. So
no, I think it makes perfect sense, right? Especially so my experience has mostly been kind of what I would call small biotech and not big pharma. So I can kind of only speak to that, right? But if you look at the expertise of people that are traditionally gathered to form small biotech teams, there's no one in there that comes from like Amazon or UPS. They're all people that were like, you need your chemist, you need your computational chemist, you need your pharmacokineticist, you need your biologist, like the traditional hard science disciplines, which we think of underlying drug discovery, and they are. And then you have a BD person, right? Because someone needs to raise the money and someone needs to sell the asset when you're done. So that person's always there making sure the runway is there and the big picture budgets. But what we don't have is anyone that really focuses on like operational inefficiencies. And that goes to exactly what you're talking about. And they are there, and the people in the room that experience them and get frustrated by them are not the ones with the skill sets or the tools to help solve them. So it's actually a really interesting opinion. Like, I totally agree. Yeah, that essentially you could reduce cycle times just by doing things that maybe aren't sexy, but just understanding and mapping, like, how's your operation?
How is this operation actually working? I've never really thought about a lot, that about a lot, or done that a lot, at any company I've been at. But like, to me, it's how many iterations do you get as well, and over time that might build up. That, hey, if it takes 4 days instead of 5, suddenly you're doing quite a bit more science there. But
I think there's a lot of hidden value to be extracted by eliminating those operational inefficiencies. The information stuff we're probably better at now, like organizing and reprocessing information.
Oh, that's a good point.
That used to be a big bottleneck. I see that getting much better, just with new dashboards and like, how like and AI helps a lot with that, but just integrating a lot of data points so that they're synthesized and integrated in a place that people can see across them. Because that used to be very spread out, and then you'd open up something, an Excel sheet from one person, but you have to open, those dashboards. I'm using that as a catch-all term. I think that is certainly improved.
Sure, yeah.
But integrating the whole process still needs a lot, right? Like, and so it'd be interesting to see. I'm sure people are working on that. I think there's a lot to be in the near term. You could derive a lot of value from getting rid of those inefficiencies. And time is the ultimate parameter you got to worry about.
It sounds like you're going to run out of it. You can run out of money. You can always fill more money, but time, the field moves off, and you're like, or some major clinical trial reads out that invalidates your thesis or something. Suddenly you're, you're kind of dead in the water. So I've just been kind of like thinking about things, how do you improve it even further? But it's going to be cross-discipline for sure, right? I mean, there is literally a logistics aspect of getting a molecule or a peptide or something synthesized and getting it to the place where it's tested. Fundamentally, machine learning, right, is feedback and iteration. So how do you reduce that feedback time where you got something new, you want to test it, and then look at that data and then iterate, make your change, and just boil that down to steps and then figure out where you can save a day. I think you're right. That's probably something that could use some improvement in the biotech field. Absolutely. Is there any other topics that we haven't covered so far that you're interested in discussing?
Yeah, I would, kind of as a broad catch-all, like for our first meeting here, really like autoimmunity is obviously my passion. It's the thing that I think about most deeply therapeutically. And I've seen immunology really move from broad spectrum immunosuppressives. I've been here for what I call the precision immunology revolution, which is like understanding specifics, usually cytokines or cellular markers that, when modulated, usually by an antibody, sometimes by a small molecule, or now by a peptide, have a therapeutic effect. But a lot of times, like, there's still never going to be that magic bullet. That there's going to be a large percentage of your population that's refractory or becomes refractory to a certain therapy. This could be your anti-TNFs, it could be your anti-IL-17s, it could be anti-IL-23s, go on and on down the road. So I think the whole idea of the immune reset, just to bring it back to your concept of adaptive T cells and CAR T cell therapy, is something I'm really, really interested to see, because it makes a lot of sense to me that a lot of the precision immunology we do just deals with symptoms, or at least, kind of just
mitigating the symptoms rather than getting to what is the underlying driver of the actual disease.
Right. Or we're one level down in the pathology, in the pathophysiology. So yes, the IL-17 is causing like the development of Th17 cells, which are causing your psoriasis. That's true.
Sure.
But what's causing you to have an aberrant IL-17 response in the first place? And it's something that's deeper than that.
Interesting.
Yeah, it's these long-lived memory cells that, for whatever stochastic reason, your genetics or your environment or the interaction of the two developed in you, but now they're deeply seated in your immune system, both physically and kind of like existentially. So every time, and they're making too much IL-17, and so you're adding a drug to block that signal, and it works. It's therapeutic. It prevents your outbreaks, your plaques. But as soon as the drug comes off, the symptoms come back. So it's like it's always boiling underneath the surface, right? And the whole idea of an immune reset, I think, is really, really interesting and powerful.
So you can wipe out the memory T cells, wipe them out, and then suddenly there's no more, aberrant IL-17 produced, because hey, we just completely reset the immune system, kind of.
Yeah. And we thought we did that 10 years. Like, we started thinking about this a long time ago, and I'm thinking about for B cells, and there's also some T cell depleters, but mostly B cells. The T cell depleters, they're almost always oncology drugs that are repurposed, which is great, right? Because you have B cell or T cell cancers, blood cancers. So we come up with these antibodies that remove those cells, but they're using markers that are on your healthy cells too. But now immunologists have this tool.
Oh, you're talking like specific. Yeah. So you're talking about specifically removing that rather than just the entire B cell population. You're moving. Interesting.
So yeah, you can either think about going for an antigen-specific approach, so just very really precision B cells, but also just repurposing those broad B cell depletion drugs. The first wave of them, I think, what we found though is when you use an antibody, it seems to deplete all of your B cells, but that's actually just because you're only sampling from your peripheral blood compartment. You're taking your blood, right? I mean, you can only get to so much blood from a patient, so you're taking it out of a peripheral vein. So you're not sampling like the lymph nodes, for example. So these memory cells are hiding out in compartments that are not adequately sampled. So you're looking at a graph, like, oh my God, rituximab was perfect. It just removed all the B cells.
We just depleted all of them. No, but only in the peripheral regions, not in the
So that's what I think.
They're still there. So you still have memory.
Yeah. But now the new technology, so I'm thinking of adaptive T cells, actually putting T cells, engineered T cells into your body, but also like bispecific T cell engager antibodies. So things that will cross-link an effector T cell to your target cell is a very different way to get rid of, to get cytotoxicity and get rid of that target cell. So these are things that seem to have a much more robust effect. So I think there's a lot of, I think there's a lot of promise there. There's going to be some toxicity. Everything is going to be a therapeutic index and a margin for sure.
Sure. Yeah. You're going to have to figure out how to limit the off target, or what would it be in this case? Limit the depletion of healthy B cells, I guess.
Yeah, people, it's funny. People have always worried about that, right? Like if you just use too much immune suppression, you're going to end up with opportunistic infections, and you're going to, going too far, the other side of the bookend. But I think, what we've seen, right, is honestly, we spend our last 50 million years like living in the dirt and eating like worms, so you needed all of your system. For 2026, we use a lot.
Not so much anymore, right?
Hand sanitizer.
Everybody washes their hands, hand sanitizer. We stay away from. I think COVID's probably changed a lot with that, right? Because I know
all the above. So I just don't think you need a robust immunity as you've evolved to have, which just means that you can get rid of a lot of it and live a very
unless you have kids.
Some people don't wash their hands as much. Maybe they shouldn't have like strong immunosuppressive drugs, but
absolutely. That's interesting. So you might. So then the therapeutic index could be wider because you can tamp your immune system down further with B cell depleters. That the therapeutic index might be bigger because there's not as much need to have a super strong immune system. Is that kind of what you're doing?
Yeah, exactly. We've learned like FcRn inhibitors is a great class, like to think about this. So this is the drug that really depletes all your IgGs. And this is, we've just seen like the first approved FcRn inhibitors were 2021. So we're right in the beginning of this field. And if you look at like the FDA and their response to before it was a new class of therapeutics, they were super, super worried about opportunistic infections, emergent viral infections, respiratory infections. They made them do a whole bunch of safety stuff on that. And what they found out was that worry was unfounded. They didn't know that. It was perfectly logical to think about that, but it hasn't played out. That people that are on FcRn inhibitors have 70% of their IgG antibodies removed do not really develop a lot more opportunistic infections than an average person.
Oh, interesting. I wonder, have they teased at that why that is? Is it just because, hey, there's, like you were saying, it's just, you know, there's not as much exposure to any of these pathogens as there was historically, or is it just, hey, there's a certain level that if as long as you maintain, you get strong immune responses, but anything below that, you might get these opportunistic infections? Is it kind of like an all or nothing, or is it a?
No, I think I think it's actually all of the above. And the third one is just the immune system is very functionally redundant. So there's that aspect of it too, just the robustness that something else is covering you. You know, that's not, it doesn't necessarily your IgGs. And a lot of times with autoimmune diseases, although they do seem in themselves like very difficult to treat, there's still, you know, a system that is out of whack, that is out of balance. And so restoring that balance is still maybe easier. So getting rid of those extra IgGs is more likely to restore balance than it is to throw off the whole system.
Yeah. So maybe there's multiple pathways that are compensating to bring it back into balance if you get, you know.
Interesting. For FcRn inhibitors, you still have your IgMs and your IgEs and your IgAs. So you still have other classes of antibodies that are covering those worries about covering opportunistic infection, for example. To your point, you're still maintaining some level of them, 30% still there. Maybe those are just having the effect. So, you know, all of the above. And I don't think we'll ever exactly know. But what's good to see.
Yeah, it's so complicated. Like immunology is one of the fields that, you know, like makes our brain hurt. It's so complicated. And you get, like I was mentioning at the beginning, and the exception is the rule, actually. And then, you know, that's why to me it's been so fascinating to work in and learn about, you know, all the different, you know, exceptions, I guess.
And there's no shortage of like new hypotheses that I think are therapeutically valid, which is really, we're still in our infancy of understanding immunology for sure.
Sure. Yeah. So there's probably a lot to learn, which is crazy to me when you start learning. You know, I took like a crash course in immunology, I remember, for chemists, and you know, it seemed pretty comprehensive, but you know, to hear that is just like that's pretty, it shows you how, you know, evolved it is, and how there's got to be feedback loops, there's got to be compensatory mechanisms, there's got to be a lot that we don't understand, kind of thing, even now.
It's a perfect example of that was the Dunning-Kruger curve, which is the more you learn, the more you know you don't know.
Sure, yeah, absolutely.
I'm very much in that immunology where I'm like, I'm humbled because I've learned so much. I completely don't understand how much I actually don't understand. So to your point.
Oh wow, yeah, very cool. Maybe it's good to end there on this Friday afternoon. What do you think?
Absolutely, man. This was fun.
Yeah, absolutely. I really enjoyed having you on, Jamie, as always. You know, it's always great to catch up, and you know, definitely like to, I always end up learning something when I'm talking with you about immunology. So it's very a pleasure to you know, work together, and you know, definitely great to have you on. So
absolutely, man. I appreciate the opportunity.
Thanks, Jamie. Have a great weekend.
All right, man. You too. Take care.